So today, what we will try to do is have pretty much the whole day practice. I'm actually going to show my laptop. So we are starting with a brief introduction quick brief introduction on Maxent, and then I'll go over the Maxent interface, and then you get to practice. And then Xiao takes over pretty much for the rest of the day, so that you can see and practice data formatting in R and running models in R. So today is a day of hands-on. If you don't have hands up, it means you know everything. And it means that it was a waste of time for you to come to this tutorial. So please have hands up when we do hands-on, because otherwise, or it may mean that you are following the guidelines and everything is going well, so that's fine too. You don't have to have a hand up, but please, what I'm trying to emphasize is work either with using our data or data that you brought, but do practice, because if you just watch us do things, it's probably not as good of a learning experience as you actually trying to run Maxent or run code. So that being said, don't be afraid to, those of you who never used ArcGIS or R, if you have very basic question, how do I display a point shape file? That's fine. There's no question that is, I don't know, not welcome. So feel free to ask us questions and do try to work on your own, because I think that's the best way of learning how to run models. Okay, so just a few slides to orient ourselves. So it's a brief introduction. I wanted to show you, first of all, where to get the download the software or the application, the Java interface of Maxent, and then I'm pointing you to, I'm citing or showing you the first paper that introduced Maxent, but there are additional papers follow up as the authors changed settings in Maxent, added options and such. They kept updating and publishing, but that's the original publication, or but the publication that introduced Maxent and maximum entropy to the ecological nature modeling or species distribution modeling world. So that paper got published in 2006, and that paper has been cited, this is Web of Science, has been cited almost 5,000 times, and it's still going up. And Town was telling me just now that Google Scholar shows
8,500 and accumulating at 1,000 per year.
So we are all Maxenting the hell out of data. And why do we do that? Why is it so popular? Well, I have my hypothesis why it's popular. It's because it's easy to use. It's because data, presence data and environmental data have become more and more and more easily accessible and available. So that's access to data, public data, that's one reason. But this idea that you have an algorithm that can use presence and background to give you an estimate of potential distribution has been quite appealing for very diverse fields of research. So the question is, where are species found, or which pixels on the landscape are occupied by the species? So that's generally, broadly, the question we are after when we study species distributions. So we can approach this question by having a probability of each pixel being occupied, which means we don't have zeros and ones, but we have a very, very small probability versus a very high probability, but all pixels, all cells on our landscape have a probability of species being present or occupied. And so basically we are using probabilities to address this, or to try to answer this question, where are species present? So that's the general approach, and it's the maximum entropy principle. Remember, Lou Gross gave a talk yesterday, introductory talk to NIMBioS, and he mentioned using Maxent, and he showed you a paper, not Maxent, using maximum entropy principle, and showed you a paper from the, I can't remember if it was 60s or 70s, but yes, maximum entropy principle has been around since, I think, 1957. I was trying to figure out some time ago. I haven't tried recently. I was trying to figure out where did it all start, and it looked like it was around 1957 that the maximum entropy principle started to be used. So with the maximum entropy principle, we determine what we know, and those are called constraints. And then we select, but the maximum entropy principle is find solutions that meet the constraints, but of those possible solutions, we choose the distribution that is most spread out. So that is the lingo of maximum entropy. The important point is to think about, to remember these, determine what you know, and these are the constraints. Okay, so in Maxent, in not maximum entropy, but Maxent vocabulary, determine what you know, the constraints are, we determine the constraints based on presence data and environmental data, environmental conditions at those presence locations. So this is, I think I took this from an online tutorial that Rob Anderson posted. So it's a very simple depiction of what's happening, but we have presence points in environmental space, and but Maxent finds those constraints in this environmental space based on presence data. So in this case, one constraint could be the mean precipitation and temperature of those points, of those locations that represent species presence. Okay, so you'll see in Maxent, you'll see these constraints as features. That's why I should wear this, not hang this. You will see these constraints labeled as features in Maxent. So we have linear, quadratic, product, threshold, hinge features, which are, again, constraints in maximum entropy language. And so variable itself, squared, product of two variables, so on and so forth. So this is all that happens internally. We provide Maxent with a set of occurrences and a set of environmental variables, and then Maxent calculates or finds these constraints based on what we know, based on this data we have available. Okay, so it calculates, the algorithm calculates the probability distribution of maximum entropy, so the most spread distribution, given those constraints, constraints that were built based on occurrence data and environmental variables. So we have a statistical model given those constraints. And the statistical model implemented in Maxent is the Gibbs probability distribution. And that's the equation for Gibbs probability distribution. So we have pixels, vectors of coefficients that are weights for those features in Maxent, which again, features in Maxent means constraints in maximum entropy. And then we have a vector of all features or all constraints that Maxent is using to bound the potential distribution of the species. So what this means is that we have, for each pixel, remember I said, we are dealing with the question is where are species present, or which pixels are occupied by the species, and I said we deal that in terms of probability. So that means that each pixel will have a probability, and it will be a tiny, tiny, tiny probability for the locations that are not present. And then in the Maxent implementation, all these probabilities across your study area will sum to 1, which means when you run Maxent, we'll see that in a minute, you have various options of outputs. The raw output is basically this Gibbs probability distribution, the values of this statistical model, Gibbs probability distribution, across entire study area, but they will be very, very small because across the study area, if you have 2 million pixels, they will add up to 1. Nobody works with the raw output, with the raw Gibbs probability distributions, because they are so tiny and because there are probably hundreds of thousands of values across a regional or continental scale. But so that's what we have. We have constraints. We have a statistical model that is this Gibbs probability distribution that is used. It's not an ecological model here. This is a general probability model that is used not just by ecologists and biologists. Okay, but on top of that, we have Maxent has a regularization parameter which limits it's an additional limit to that most spread distribution. So we have the constraints that limit the distribution, and then we have a regularization parameter that also limits the extent of that maximum entropy. So again, if we have this simple depiction, precipitation, temperature, our presence is in red. We have the sample average, but then we are searching for the maximum most spread distribution given these constraints, the features, and then the regularization parameters. So the sample average may be this, the true mean may be that, and then Maxent creates limits to how far we go in terms of this maximum entropy. So basically the regularization parameter decides how spread the distribution will be. And you'll see this, we'll play with the regularization parameter in Maxent. And I think that's the last one I have, which means we are gonna start Maxent. So this would be a good point to ask you if you have Maxent on your laptops, and if you have the environmental data that was posted on WordPress, or if you don't, if you want to use your own data, that's fine, but it would be good to have data now. The one limitation is that my computer has the screen has high resolution, and everything is very, very small print, so it's hard to read what I'm doing. I'm going to try to see if Maxent was installed on this computer. I don't see it. Okay, so we'll go back to my computer. Okay, so if you downloaded data, how many of you are going to use the armadillo data that... Okay, so there are a few. Okay, good. So there are a few. So it makes sense to check those data. So last night I downloaded the data to see what it's like, especially the presence data. So I downloaded the data and I mapped it. The first step: map your data, see what you have. And there was one point that was clearly not in North or South America, and I couldn't figure out if it was flip of latitude and longitude, and I decided to just eliminate that one point. But there are, I think, thousands of points, so one location I eliminated it. My computer is taking forever to... That's why people use R and QGIS. Okay, finally. Okay, so
I have a question.
yes, please.
So I also come from the occupancy world.
Mm-hmm.
So I'm having a little bit of trouble understanding what really this probability of a pixel being occupied is. Because
Yes.
So in occupancy, you are arguably modeling the source of all zero data. So here, I mean, that probability is really the probability of a pixel being occupied by the species?
It's my interpretation is that it's not. So, but it's an interpretation coming from a non-mathematical, non-maximum entropy, and so it is probably less informed. But so my interpretation is this: we have occurrence data, we have locations of presence. We don't have locations of abundance or prevalence or anything like that. We have, yes, I saw the species between 1950 and 1990 or 2000. I saw the species one time here in this pixel. So the algorithm is creating those constraints based on locations of presence, environmental data, and of course the background, contrasting the presence with the background. My interpretation is that these values represent similarity of environmental variables, similarity across the landscape to those training presences. So I don't call it presence, or I can't remember what is the other very used, commonly used interpretation of Maxent outputs. But yes, probability of presence implies a lot more than the kinds of data that we provide. And please use the microphone.
Nope. Yes, there we go. Just a quick comment. If you read the Phillips papers about kind of the derivation and justification for Maxent, they do try to spin it as a probability of something. It's a pretty convoluted terminology for what it's a probability of. My conclusion from reading those papers is that the set of circumstances under which you could consider it as a probability is so restrictive that it's better not to consider it a probability of anything. So what you'll see with the methods that I use is that I'm always just trying to take it as a monotonically increasing index of suitability. And so I kind of, don't make any interpretation of the difference between, 0.1 higher probability or 0.01 higher probability. It's just, is it higher? Is it lower? So again, my advice would be to interpret Maxent outputs simply as an index of suitability, nothing more.
Yeah, I think, predicated on the data. I meant to, I don't know how, I feel like we only have half hour and I should show you all the settings in Maxent because there are so many. But I did want to mention this in relation to what we'll see in the Maxent interface. There is a paper that was published last year. The title is very encouraging, Opening the Black Box. So Maxent, for a long time, the code was not open source. It became open source before 2017, but this paper, particularly the most recent paper by Phillips, talks about an additional output. So there is a raw output, there is a cumulative output. These were the first two ones released in 2006. Then I think 2008, the logistic output came around, and now 2017, we have a C log output. That C log output has to do with the attempt of converting these Gibbs probability distribution values into Poisson point probabilities. And we talked a little bit yesterday about what is Poisson point, and it has to do with intensity of points or intensity of data that we have. and it's predicting intensity of data. But so this C-log-log is attempting to do that, or this is the proposed output for to get close to this Poisson point process. But, and I'm trying to find that particular, there's always caveat. Somewhere in this paper, don't want to spend too much time, there is a mention on how presences are, the assumption is that presences are independent. So if you have a flock of birds, or if you have social mammals, or if you have, I don't know, think about that, aspects that make species, individuals' presence not be independent. So if you are in a situation of non-independent presences and background, they say, then you can't really use that C-log-log. So it's a long story about all these outputs that are modification of that raw Gibbs probability value, but that Gibbs probability value is not probability of presence, it's probability of similarity of environmental variables, conditions, or what Town said, probability of suitability, because suitability is basically environmental conditions being similar to the presence data. So Gibbs probability values represent the raw values represent some approximation of suitability. Then there are transformations, cumulative output, logistic output, and now C-log-log output. These are all transformations attempting to get from these Gibbs probability values to something that is more akin to presence, and in this case more akin to Poisson point process. But all these transformations have a lot of assumptions about data. And if you have good data, maybe you can say, yes, these values do actually approximate quite well probability of presence. If you throw just GBIF data at it, I don't think you're in that situation. However, we'll see that all these possible ways of expressing Gibbs probability distribution values don't actually matter once you threshold the model. So once you go from this continuous output to presence absence, it doesn't matter that much. The variation, the small variation, the small increase or decrease change in that probability value, it becomes just 0 and 1. So do read this paper. If you're going to use the C-log-log, the latest output, because that is now the default output when you run Maxent in the Java GUI. And so you do want to understand what this most recent output is. But like I said, it depends on intensity of sampling. So if I just have presence and I don't have abundance, I don't have multiple sampling of the same pixel across the landscape, this kind of multiple sampling, this Poisson point process is not going to work for that situation. Okay. Anything else? It is confusing. We could spend the next, week talking about Maxent. And I remember when the paper came out in 2006, I must have read that paper 6-7 times, and I kept finding new tidbits that I didn't realize that were very important for understanding the output. Okay. So back to the data, that armadillo dasypus data. So what Xiao uploaded is, let's take a quick look. Are you gonna do? You're probably gonna show the presence data when you do. Okay. So what Xiao uploaded is a file that you download. I mean the format, the complex structure of data with lots of fields. But I removed that, and I have only latitude and longitude. So I'm just adding that, a slightly modified version of the original dasypus occurrence data, which has this file has only latitude and longitude. So remember, you add the CSV file, and then you display XY data, or you can go to File, add XY data. So now we can see those points. So I remember there was a point somewhere here, and I deleted that point. You'll probably see it. It doesn't really matter because the environmental data will be for North and South America. Now, this is a species that has slowly shifting its northern limit into US, farther and farther north in US. So this is slowly extending distribution, meaning that at this at the northern edge of the distribution, we are not dealing with species in equilibrium with the environment. So the first problem is that we would have to make some decisions where we draw the line in terms of the northern limit of the species. So I'm not doing that for this, just showing you Maxent. And then we do have these points that are not part of the main distribution of the species, but I'm keeping them in for the purpose of just showing you how to run Maxent. Other problems, you have extremely dense sampling here, but we just want to see Maxent settings and outputs. And then the other thing is we need a study area extent, right? So we have occurrence data, now we need a study area extent. And what I did was just that simple polygon approach buffered by, I think, 100 kilometers. So now this will be my, again, this was late last night. We need some, demonstration data to show how to run Maxent. It's not a biologically informed extent of the study area. It's basically an extent of the study area dependent on knowledge of presence of the species. Okay, so that's that. Then I have occurrence data in this form. Uh-oh, where's the table? Oh, here. Right, so I need presence data in this form for Maxent. I need a study extent, and then I need the climatic variable to be, or environmental variable to be clipped at this extent. So I did the clipping last night. Of course, we're not going to do it right now, but we have that handout on how to clip the climate data to this extent. The other thing is the data set that we have uploaded, and do not look at date modified. I just realized that. Never mind. So we have that that World Clim data set that has 19 bioclimatic variables, and I showed you yesterday the website that you go to. And I'll show it again because we show lots of things and you probably don't remember. Right, so these 19 variables, these are the labels for the 19 variables. So you have annual means of precipitation and temperature, and then you have seasonal extremes of precipitation and temperature. And by seasonal we mean 3 months averages. Okay, so that's that. So basically, I have I have 19 variables that will be clipped to this that I clip to this extent. And what I'm not addressing right now is the fact that I have 19 variables, right? And we talked yesterday about multidimensionality of the environmental space, that many of these variables are correlated, many of these variables don't make sense are not driving the distribution of the species. So if I use ecological or biological knowledge, I will remove some of this variable. If I do a correlation analysis, which I showed you in SDM Toolbox, I will get just a subset of the variables that are not correlated. And then third, what I can do is another option that has been used in recent literature, or not necessarily recent, but in the literature, is run a Maxent model, and then Maxent will rank the most important variables, or the variables that contribute most to increase in model fit. It's called gain. And then you run a second model with just those variables that contributed most, and we'll see that output contributed most to the model. So there are three options. I decide based on biology, I decide based on correlation, or I let Maxent figure out which are the most important variables. And you may find yourself in a situation where Maxent ranks two variables as the most important, and they are correlated because you are getting collinearity in your data set. So again, we are just running, just going to run a Maxent, and then we are going to look at the contribution of variables. So this is where having a high resolution screen is very limiting. And that is, you can't really tell, you can't really read, but here it says samples, and then here it says environmental layers. So this area is for loading your samples or your species presence data, and this is the area where you load the environmental layers. And there are a bunch of other options there, and we'll talk in a minute. Okay, so I'm gonna go load the data, and I need a magnifying glass for this step. Okay, so I have just one species in that CSV file, and it's listing just that one species. And I used just the genus name. I didn't use the specific epithet. But if you want to model 5, 10, 100 species, what you do is that spreadsheet, that text file, or CSV file, not text file, CSV file will have species 1, latitude, longitude, 100 records. And then species 2, 50 records, the next 50 rows, latitude, longitude. So you will have your entire data set of presences for all species in one CSV file, species 1, species 2, species 3. If you have that, you'll see species 1, species 2, species 3. And then you can run all models at the same time for all species. Yes? Oh, I have, so what you have on WordPress is, you have on WordPress, you have this. Oh, I should have double clicked. So you have the raw data. Yes. Sorry, yes. Non-mammalogists, well, yes. That's the genus name of armadillo. So Dasypus is armadillo. So that's the data set that is on WordPress. And then remember I said that if you try to open it in Excel, you'll probably have problems unless you do from text CSV. So data from text CSV. And then so this is the original data that has all the fields. And then from that original data, it's about 5,000 records, so it's taking a while. It comes by default with this filter. Ooh. Okay, anyway, so from this Darwin core output, what I'm using is just the lat long, just the two fields, latitude and longitude, or wherever they are, here. So here is longitude, and latitude is somewhere in here, or over here. So basically, I just copied those two fields, the latitude, longitude. I did what what we shouldn't be doing, which is just grab the data that you have available to run models. But yeah, so the latitude, longitude are in this big file here. So that's that. And I just did, yes, I just saved the two. No, I did this at 1:36 a.m. No, because I did it at 1:36 a.m. Yes. But so the point. So the point, what I'm trying to do right now, if you look in the schedule, what I'm trying to do right now, let's go to the schedule, because I understand it's confusing. So this is what I'm doing. Basic settings, advanced settings, Maxent outputs. You are practicing after I stop talking. So please pay attention. So after I stop talking, then there's practice Maxent. So what I'm trying to do right now is show you all the settings in Maxent. You don't have to follow click, 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 but then you have to follow once I stop talking, and I should have stopped talking a long time ago. Okay, so, but anyways, yeah. Okay, so back to this interface. So we load presence data here, which is species, and then longitude, latitude, and then environmental data. And the reason I'm saying follow this is because you will have to process environmental data. So it's easy to copy longitude, latitude, and get a species presence data file right now, but you would have to do this while I'm talking. So meaning this, you'd have to generate the extent of study area, and then you'd have to follow that handout of processing raster data right now. So don't do that right now, because it takes time, and I want you to pay attention to Maxent. We are doing Maxent, and then you'll do practice after we go over this. Okay, so I have presence data, and then I loaded the environmental data, which are those WorldClim, BioClim layers that I clipped to the extent, to that polygon extent, and then I saved them as ASCII files, because Maxent works with ASCII files, not with Esri grids or GeoTIFFs or anything like that. It requires ASCII files, which is why we have that in the handout, how to save ASCII files. Okay, so what we see here are the 19 variables, and you can click and unclick. You can, not use 19 variables. You can use your 5 variables that are biologically meaningful. So that's that. Okay, then we have some boxes here. One says create response curves. So I'm going to click that box, create response curves, because that gives you the variation in environmental space, one-dimensional, and relationship to probability, Gibbs probability or suitability probability. So we'll see those response curves, and then if it's a flat response curve, it means that no matter how much variation is in the temperature, the probability of suitability doesn't change. So it's not a factor that influences that species distribution. So that's the create response curves. Make predictions of, make pictures of predictions. You can leave that clicked. It's just going to be a PNG, a little PNG figure. And then do jackknife to measure variable importance. This takes a bit of time. I'm not going to click that. I'm going to, because I want to see the results fast. But if you click that box, what will happen is there will be a jackknife implementation where a variable is left out and the model is run. Then a variable is used to run a single model. So then you will get an in the HTML output, which we'll look at, you will have another section. I won't have that, but you'd have another section that shows how the model performance, so area under the curve, AUC, how that index or how that metric will change as a variable is eliminated from the set of variables, or how that one particular variable, how important that one particular variable is when you run the model with just that variable. So I'm not going to do the jackknife procedure because it takes a while. And then here's the box that says output format. And now you see that the output format, the default is C log, which is that attempt to get intensity of points or presence that Poisson point process. I'm going to do logistic output, which is a modification of the raw, and that is an attempt to get closer to, basically to normalize over those raw probability values that are tiny, tiny, tiny. So I'm going to do logistic. You also have cumulative. And if you want to see raw, there is raw as well. Cumulative is an older one. We can talk about it. It's not very it's not a very good modification of the raw ones because it's counterintuitive. The lower the cumulative value, the better the prediction is because it includes more of the presence data. So we're not doing that cumulative. We are doing just the logistic. Then the next box is output directory. So this is where the model outputs will be saved. So I'm just going to create a folder. Results. Okay. So all the results will be saved in this new folder. And then the next box says projection layers directory file. So here is, in this box, in the projection layers directory, you can load environmental data that represents a different geographic region or represents a different time frame, thus different climates. So, in other words, if I work with an invasive species, I train the model with these presence data, these environmental variables. Maxent generates a model, an estimation of the niche with these two data sets, and then it takes that model and it projects it to another geographic region. So let's say it's an invasive species that is native in Europe and has been introduced to US. I'll have training data from Europe, and then I'll project the model to the same environmental layers for US. If you do climate change projections, same thing. You train the model in the present and you project the model on future data sets. They have to have the same, the future layers have to be the exact same as the ones you are using to train the model, and they have to have the exact same name that you are using in the training. So if I have temperature, I cannot put here temperature 2050 for future. The layers have to have the exact same name because it's looking for the same variables in the future, in the projection data set. So that's that. Then here, you have those constraints, the maximum entropy constraints. So you have linear, and again, in Maxent, they are called features, but they are constraints. So you have linear, quadratic. You see that the default is auto features, and that means that Maxent, if you leave auto features, Maxent will decide how complex the model will be. The more features we include, the more complex the model is. And Maxent will decide, the algorithm will decide how complex your model can be based on how many presences you have. So if you have only 20 presences, it will actually do just linear. With auto feature, it will do just linear, it will use just linear constraints, just variables themselves. If you have, I can't remember what's the threshold for including quadratic features. If you have more occurrences, it will use quadratic features. If you have, I think, more than 80 occurrences, it will use all these features. So if you leave auto features, it will decide how complex the model will be based on your presence data. If you decide not to do this, if you decide to manually choose the features, the constraints, obviously you don't check the auto features, and then you can check and uncheck these features. So I go with auto features generally because of optimization of the model done with about 250 species, with ranges varying from global to endemic species. So this auto features has been optimized using a large number of species and a large distribution of data. So I usually go with auto features if the models are overfit, meaning the omission error is high. Then you can go in here and usually if you remove the threshold and the hinge features, you're starting to get less overfitting models. And sometimes if you look at the threshold and hinge features, it's a bit biologically, they don't make that much sense because you have a threshold, and then above that or below that above a threshold, it becomes a constant value. So some of these, the threshold and hinge features biologically don't make that much sense. But they are used only if you have lots and lots of data. Yes?
So I have so many questions.
Go ahead.
As for letting Maxent decide which variables are showing the highest impact on the response, would you be, so the variance inflation issue, so in
Yeah.
NIMBioS, you may lose the signal if you put correlated variables, but
Yes.
also you may get in bias estimates because
Yes.
the information is so redundant that then it's garbage.
Yes.
So here we can have the same issue.
Yes.
Not only that you don't know what is really driving the thing, but also the output is being biased by this redundancy in everything.
Yes. So the default approach that I have is to run a correlation analysis and remove highly correlated variables. But with a couple of studies, a couple of projects where we played with removing variables based on correlation, removing variables based on biological non-significance, and allowing Maxent to do whatever it thinks it's best, we don't see big changes in the AUC omission error. And Xiao can tell you a lot about that because he ran a lot of simulations. So it is an important, what I'm trying to say is that I've been freaking out about, I never use 19 variables and I do not want you to use 19 variables. What I'm saying is that you can use one of the three options to remove, not to fit your model with 19 variables. In the instances we tried, the differences are not that big. But you may have species that are very dependent on two variables, and the rest of the variables are garbage and noise in the system.
Or a stepwise approach, or like what's the?
It's not doing stepwise approach. It's running iterations, and then it's ranking the variables based on how well a certain combination of, it's not just variables, unfortunately, it's transformation of the variables. So it's a combination of variables that are used and transformations of the variables that are used. So those combinations are run in an iterative process, and then the models that have the best fit are then kept. And so what you see in the, what we will hopefully eventually see in the model output is a ranking of the variables based on all those iterations down.
Sorry, just to give you one set of options. I totally agree with the idea of reducing by high correlations, because those are essentially copies of environmental variables. So you can reduce complexity somewhat. But then we get into essentially where it becomes a little bit by art, you know, by what we know and what we believe. So our package that we'll show you at the end of the day, which is not yet submitted for publication but will be soon, you are able to set it up so that it will look at all combinations of the environmental variables that you give it and all combinations of the feature types. And so you know, if you've got the time and a fast enough computer, you can have it cycle through thousands of candidate models and pick out the optimal balance. And one thing that we've noticed is that sometimes you'll get fewer environmental parameters, but a lower value of the beta parameter, which I don't think you've talked about yet, which is to say more complex models based on fewer environmental parameters, or simpler models based on more environmental parameters. So as we've started exploring these, you know, these exercises where we look at thousands of models, we've found some interesting kind of disjunct islands of possible solutions. But we'll look at those outputs tomorrow more than anything.
Yeah, but definitely don't just run and the other thing is, you need to have lots and lots of presence data if you're going to have a highly complex environmental data set. If I have 19 variables, I need to have probably at least 100 occurrences or something like that. And a lot of times we see low sampling size, high dimensionality of the environment, and we know that is not a good approach. Okay, so environmental data, presence data, environmental data, response curves, pictures of predictions. We can do jackknifing of environmental data. Then the output type, the directory where where results go, and then projection if you're doing future, past projections. If you're doing invasive species, if you're projecting from one area to another, that's where it goes. Features, I would leave them as auto unless the results, the initial models look strange. So pretty much we're ready to hit run. But if we click on this button, settings, we open a whole or multiple cans of worms. Because I hit settings here, and all of a sudden I have 3 tabs here. So the basic tab allows me to do several things. I can select a random seed, meaning the presence data set will be randomly subset into training and testing. So this random seed is coupled with random test percentage. So if I choose random seed, then I will choose a percentage, so let's say 30. And I will use 30% of my presence data as a test data set. So after the models are run, then I test the models with 30% of the all presence data that I have available. Other things, give visual warnings if there are problems, show tooltips, which is this text box that tells me what each of these options are. Ask before overwriting, skip if output exists, remove duplicate presence records, and I have that clicked, because that's the lazy Mona approach. Then write clamp grid when projecting and do mesh analysis when projecting. So these 2 options are for when you use a set of projection layers. So when you, again, project your model future, past, another geographic region, you will have 2 additional outputs, which are a clamp grid and a mesh analysis. And mesh stands for multivariate environmental similarity surface. And what that analysis does is it's figuring out in a multivariate space which pixels in your projection data set, climate data set, and again, projection can be future or can be another geographic region, same time frame. That mesh analysis is doing a multivariate approach to identifying pixels that are different in your projection versus training. And I'll stop at that. There's a whole paper about this mesh analysis, and there's a whole follow-up paper about how, or at least one that I'm aware of, about how this mesh analysis is messy, and we should mop the mess. There's a paper the official title is not Mop the Mess. I think, was it SOAP, Erin? Oh, even better, yeah. So I won't say more about the mesh analysis. It is an interesting output. It has limitations. Read the illiterate paper about the mesh analysis and the follow-up, mopping the mess, SOAP et al. 2014, maybe? 12. Okay. And we could spend another couple of days or more talking about these projections. There are immense, not immense, but there are several potentially dangerous pitfalls with projections. You have, a type of relation or a space of relationships between environmental variables in the training region. Once you project it to a different set of environmental data sets, that correlation matrix will be completely different. So it's messy. But so you have this option of looking at an output that tells you which areas of the projection are environmentally different, and that is important because that means the model is extrapolating outside of its bounds, training region bounds. So those pixels that are predicted present or absent in that space that is not within the bounds of the training region, so that projection space is basically questionable. The model is extrapolating beyond the data that were used to train the model. And then you have also related to projections, right, clamp grid. So Maxent is trying to not extrapolate outside of the bounds of the training environment by creating a clamp grid. And you can see the clamp grid. You can see the areas that are outside of the training, the environmental space bounds. And you can identify those areas as problematic. Okay, so these two are for projecting the models. I already talked about random test percentage, which is I put 30% of my data, of my presence data, to be used to test the models. You could put 50%, 25%. It's not a very rigorous way of doing this, which is why generally we don't want to just run a Maxent model with 30%, with a percentage of the data used as training. We should be, what we could be or should be doing is using different percentages, or even better, using this option, which is creating replicates. So you can create replicates in a cross-validation approach, bootstrap approach, or subsample approach. I'm a big proponent of cross-validation, especially when we have small sample sizes, because then instead of just one time separating, I have 20 presences, I'm going to use 5 presences to test the model and 15 presences to train the model. Instead of doing just one split one time, I'm using multiple, I'm using 5, if I count well, replicates, and each replicate is using an individual set of presences to train the model, and then an individual set of presences to test the model. And then the next replicate will use a different set of presences to train the model and a different set of presences to test the model. In other words, I will use a different subset every time. I'm trying to think how to illustrate this. I will use, it's basically without replacement. I will use, if I have 10 occurrences, the first replicate will use these 3 to test the model and these 7 to train the model. The next replicate may use these 3 to test the model and these other 7 to train the model. So each replicate is using each point once to train or test the model. So that's without bootstrap with and without, what am I trying to say? Basically without overlap between the points that are used for training and testing. The word is not replacement, thank you, without replacement. So that's cross-validation. I am a big proponent, but the reality is that it works. I think it's good when you have 10, 20 occurrences. I think it's a good way of dealing with low sample sizes, but it's not, I don't think statistically, I don't think it's a very strong approach when you have 1,000 occurrences, when you have large data sets. I am still inclined to use cross-validation a lot of times. So that's cross-validation, then you can do, so if I do cross-validation, then I will have, let's say I will put 5 replicates. If it's a small, if I have only 20, If I have only 20 occurrences, and if I do five replicates, I will have 4 points for training, for testing, and 16 points for training. And I'll do that 5 times, like I explained here. Now, if I do this, then the random seed and the percentage don't matter anymore. So once I put cross-validation and replicates, it will do that. It will not do random seed and 30% testing and 70% training. Now, I can also do bootstrap and subsample. So bootstrap would be, well, in this case, I have 10 occurrences. I'm going to run the first replicate with 7, randomly sampled 7, and the remaining 3. And then I'm going to run the second replicate with randomly sampled 7 and testing 3. So when you have a small data set, if you do bootstrap, you could end up having pretty much the same data used for training across replicates, because you have a very small pool of data. So it works better with large data sets. Subsample, I'm trying to remember, I think it's I can't remember what subsample is doing. And there should be an explanation. I'll get back to this. I remember I played with this, and it wasn't a good, in my mind, it wasn't a good approach for what I was doing. So now because I haven't used it in probably, I don't know, a couple of years, I don't remember what subsample is. I'll get back to you about that. Okay, now, so this is all about how do you calibrate the model or train the model, and then how you test the model. And again, you can do random one-time split of data in training and testing. You can do cross-validation with multiple replicates. I'm not going to do that because that takes a bit more time, and I just want to see outputs at this point. You can also not do random cross-validation, bootstrap, subsample. You can just do your own. I'm going to load these data that I loaded as samples. I'm going to use all these data, the entire data set, to train the model, and I will add here in test sample file, I will add my own data set, my own locations with latitude and longitude that I want Maxent to use. So if I want not to rely on these sampling techniques in Maxent, I can do this. I can just load my own test data set, and it has to be the same format, species, longitude and latitude, a CSV file. So that took forever, and it's just basics. Now we get to advanced. So this is where we deal with the background sampling. And I think the add samples to background is in between iterations, because Maxent will run multiple iterations in an attempt to fit our data. It will add new samples. It will resample the background every time it does iteration. So I think that's add samples. Yes, or it's not already present in the background. So it's new combinations of environmental variables in the background. All samples is, even if the combinations are present, it's basically repeating the same combinations of environmental variables in the background. So this is repeating combinations. This is using unique combinations every time the iteration is going on and the background is sampled. Plot data, if you want to redo the plots that are presented in Maxent output, which hopefully we'll see soon, you can get the raw data that was used to make the plots. Extrapolate works for projecting on new conditions, which happens almost all the time, not just when we have actually a projection file. So the model, when you have this box extrapolate, the model will try to extrapolate beyond the bounds of environmental space that is present in the training data set. Then do clamping is for future projections. Write output grids, we all want that. We want to see the grids, the maps of potential distribution. So write output grids is, unless we are doing just some simulation, just some preliminary data, preliminary experiments, we will want to see the output grids. Plots are graphs that are present, that are included in the results file, you can have a summary of results appended to the Maxent results file of all, a summary of all results, ASCII files. Okay, now here it is maximum iteration. So by default, Maxent runs 500 iterations, meaning it will run pretty much 500 experiments within one Maxent experiment, 500 models to fit data. So by that, I mean it will play with the combination of features and the combination of environmental variables in a set of 500 iterations. And then it will stop. Or it will stop when the change between iterations, between attempts to fit data, reaches this convergence threshold of 0.501. So the model will not run forever. It will reach this convergence threshold, at which point, meaning that the iterations converge, become very similar. So if it reaches that threshold, it stops. If it hasn't reached the threshold, it will stop at 500 iterations. So it will not run forever. Sample radius would mean if you want the background to be sampled around your presence data, so limit the background, log file, default prevalence. We keep the default prevalence to 0.05, which means we are assuming that the prevalence of our data is 0.05, and I have not played with default prevalence because I don't have a good measure of prevalence in general when I use presence-only data. Applied threshold rule is for converting the Maxent map, potential distribution map, converting that to, or potential suitability map, convert that to present absence. So remember I showed you lowest presence threshold and how you can use the lowest Maxent value that will predict present all training data. And then I showed you a paper that had, I don't know, maybe 15 types of thresholding. So these are options to threshold your model. We are going to use that minimum training presence for now because that's the one, lowest presence threshold is the one that we talked about. Bias file is useful if you have that information. So if you can generate a raster that quantifies bias in your data set, in your presence data set, you should attempt to use that. So if you have information about a sampling bias, this is where you would input a raster, an ASCII raster, that gives a value for each pixel in terms of bias, high bias, or low bias, or no bias. Okay. Then we get to experimental, which is, so this is where if you are not okay with these auto features and if you don't want to unclick and click, what you can do is basically change the number of samples, the minimum number of samples at which these features are used. So in this case, we have hinge threshold. We need at least 15 occurrences to have a hinge feature included in the model. So you can play with that if you want. I would play first with auto features and change the number of constraints I have here before I change the number of presences that determine which constraints or features I'm using. Now, somewhere in here, we have the beta threshold. Wait a minute. Okay, let's go back to advanced. I'm looking for the regularization or beta. I guess this is it. No.
It might be on the first page.
First page. Oh, regularization, here it is. I skipped the regularization. How did I manage to do that? Okay, so I skipped the whole 3 columns here, sorry. Okay, so Yes, because I was focusing on how you subsample your presence data into training and testing. But these three boxes here, two boxes here. So you have a regularization multiplier and maximum number of background points. So regularization multiplier, remember I said the model, the maximum entropy is limited by constraints or these features, and then it's limited by that beta regularization parameter. So this is that regularization parameter over here. So if we have an overfit model, what we can do is go into regularization multiplier, and the default is 1, or using the optimized regularization multiplier that is included in the algorithm, or a multiplier if I put 5, then the model will become broader, so less overfit. If I have a broad model, I will go down 0.1 or 0.2 or 0.3 to get a more constrained model. So we will do this, we'll try to do this, play with the regularization parameter in this session so you see differences. And a lot of times the differences are subtle, are not. A lot of times we run multiple regularization parameters just to make sure that the model is not overfit, that the bounding box is not too limiting or not too broad. We do that and we compare the outputs, and the differences are minute. So a lot of times just the default regularization parameter works. Okay. So I forgot to mention regularization parameter, multiplier, and maximum number of background points. This is very important because the default is 10,000 samples. So what this means is I have an endemic species that is found in Smoky Mountains National Park, and I run the models with environmental data that is at, 200 meters. I will have maybe I will have 10,000 pixels. If I do the same analysis and I run it with climate data at 1 kilometer, I will probably have 300 pixels. And that's definitely not a good model. Don't do that. But just for the purpose of illustrating this, you say maximum number of background points 10,000, but if your study area does not have 10,000 pixels at the resolution at which you are running the models, it will actually use all those, it will use the entire study area because the default says the 10,000. That's one aspect of background sampling. The other aspect is if I run the models at the continental scale and I have 5 million pixels at 1 kilometer, it will use 10,000 points to do that contrasting between my 1,000 presences at the continental scale, 1,000 presences, and then 10,000 background points when the background is actually 10 million pixels. So 10,000 points will not capture very well the heterogeneity of the background if I work at large scales, large extents, not scales, large extents, 1 kilometer, or narrow extents and very high resolution of data. So, in other words, if you have millions of pixels, don't use the default 10,000, the default number of background samples, which is 10,000. If you want to compare, this is another thing, we try to compare species predictions, and if their training regions are different in extent, then we will have a model that is very well calibrated because the area was small and there were 10,000 points used to characterize the background. So I have a narrow range species, and then I have a wide range species, and I'm comparing their outputs, or I'm even projecting them to future. Those models will be calibrated on the entire heterogeneity of the background versus 1% of the background. So that's problematic when you run multiple species. It's also problematic, again, when you run a large extent species and you have heterogeneity that is not, or variation that is not included in these 10,000 points. So one approach, yes? I have not seen a paper that discusses rule of thumb. What I have done in the past has been to decide the percentage of background sample based on one of the species. So if I have a species that, based on the limitations I have across the multiple species that I'm running. So if I have a species that has a maximum of, I don't know, 10,000 points, so I will try to use a comparable percent of background sampled across species. If I have a single species, I really don't know what is. It depends on how variable, how heterogeneous your background is. So it's not, yes, another good rule of
I think it also
thumb.
depends on kind of the prevalence, the prevalence of the species across the landscape.
Yes,
If the
yeah.
species is occurring broadly across your calibration region, then a lower percentage will be enough. But if the species is very, very localized and it's in a tiny percentage of the overall panorama, then you need more background points to detect those tiny pieces.
Yeah, so if you have mourning dove and you have, I don't know, 10,000 records in North America, or you have, I don't know, an endemic, or not an endemic, but a species that is not well sampled, not so well sampled, the wolverine, and it's more limited, then you need to adjust the background sampling based on prevalence. Yes, Shao. Mm-hmm. It will. So do multiple models with the same number of background points. Okay, yeah. So if you, let's say you run 100 models, same species with 10,000 points, and you don't see a change in your model performance, then 10,000 points is okay. If you use 1,000 points, if you use 1 million points, and you have a variation across the simulations, is that what you're saying? Then you have to, basically you have to find the sample that is giving you uniform model performance across those simulations or replicates.
Basically sampling availability, correct? You know, if you're using, right, you're tracking data and you want to see what animal is using to what is available.
Mm-hmm.
Basically, the number of random points you need to generate for each present to capture what is there. So
Yes,
it's the same rationale.
I guess, yeah, I didn't think about that, but yes.
If you have a very complex topographic area, you might want a more random point than if you're working in the plains of North America or a grassland in Uruguay.
Because the signal, you can track, if you don't have topography, you can track,
Yes, if your area is less heterogeneous, maybe with less random points, you are capturing what is available.
Mm-hmm.
If you're in the Andes where things change abruptly
Yeah.
within 2 kilometers, maybe you might need more
Exactly,
points to capture what is
right.
there for the species sample.
Yeah, so it's this depends on how heterogeneous the environment is, and depends on how much sampling you actually have. The pixel, what's the pool of samples or pixels? So yeah, it's important. It stays default for now because we want to see results. I'm way past my allotted time. I think now I covered everything as fast as it was possible. Any other questions about this? Okay. So now I checked, I didn't just load data and hit run. I checked also the settings. So now let's run. So first message I get is this sample is missing some environmental data. I hit OK, and then it tells me, oh, you have another one. OK. Oh, yeah, you have another one. OK, I'm going to suppress. So those points could be coastal points that the raster is not capturing, or could be points that are in the ocean. I have not cleaned the data, and they are points, and I left them there intentionally. I saw that last at 1:30 a.m. I left them there intentionally. So it could be that you suppress the warnings because some of my 4,000 points. Sorry, some of the 4,000 points do not have are off the coast. So these are the points that are not going to be used because they are missing environmental variables. Sometimes you suppress similar warnings, and then all of a sudden the model is done, and then you realize, oh, I put latitude instead of longitude and longitude instead of latitude, and all my presence data were in Europe when the environmental data was in US. So you may get suppressed similar warnings, and in the end the model just runs very fast and there's no output. Make sure that map your data and make sure that your lat long data are in the right spot. Okay, so that was one warning. You may get, and a lot of times we get that, you may get another warning that says that an environmental variable is outside of bounding box. That happens when you say, oh, I'm going to run climate data. I'm going to use a model that has climate data. And then maybe a week later you say, oh, I found some soil data and some elevation data and topography. I'm going to add that to the model. If you use ArcMap and you don't have the exact same settings that you used to clip the data 2 weeks ago, when you try to add those 4 variables into the environmental data set that you process separately, you have to make sure that you have the exact same extent, because otherwise Maxent will not work. So let me show you what I mean by that. So it's going to be a large file, but I'm going to open an ASCII file. So this is the ASCII file in looking at it in text format instead of looking at it in ArcMap. But what I'm saying is, if you have a data set of climate variables and then sometime later you add more variables and you did not use the exact same mask, the exact same settings in ArcMap or ArcToolbox, you may have, instead of 2081 columns, you may have 2082 columns. Maxent will not like that. So you will fret over this and I had this problem, not understand what the heck is going on, and then it's just one column or one row difference. So check the header of the ASCII file if you have errors like that in Maxent where some variables or a variable is outside of that extent or the bounding box. Okay, so we have models. I mean, the Maxent finished running. Okay, so when I go into the results folder, I have this I have first an ASCII file, which is the prediction, the potential, the suitability estimate, map, or raster. And then I have an HTML file, so I'm going to open that. And this HTML file basically gives me all the results. It's a summary of everything that happened while the model was running, including variable contribution. Also, but what I find very useful is it includes what I did. So 6 months later, when I have to rerun the models, I can get back to this and figure out what I did. If I didn't take notes, figure out what I did and pick up from there. So this is a summary of what was used in Maxent, and then these are all results. I will point out the ones that are most useful, and you can read explanations, but this is the famous AUC, right? So I have an AUC of 0.8 for training and 0.84 and 8.4 for training and testing. So the test is the one that's important. So it's above 0.7. That's kind of that's the cutoff we are using for useful model versus not useful model. So this is at 0.84. It's considered useful. Then I get to this table that looks confusing, but what it is, it's showing different threshold options and how those threshold options affect my omission error and area predicted present, right? So I'm going to skip so these are cumulative thresholds, which we don't use the cumulative output. Then we get to minimum training presence and 10 percentile presence. So that means I'm allowing the model to omit, here I'm allowing the model to omit 10% of the training data. And here I'm not allowing the model will predict all my training presence data as present. So take a look at the outcome of the different thresholding technique. So if I say include all my presence data, and mind you, some of those presence data, I downloaded them from GBIF, I didn't clean them. Last night when I looked at them, there were some presence points that had just degrees and no decimals. So it was probably the centroid of the country. So if I say I need all those presences, regardless of how good or bad they are, I need all of them to be predicted present, the logistic value, the Maxent suitability value is 0.002, but so that's the Maxent value at which all the pixels are predicted present. At that value, 97% of my extent, 97% of the Americas, or South America and part of North America, 97% is predicted present. Test omission error, so all my presence data that were the 30% that I left aside for test, all of them are predicted present. But I have 97% of the, I have a high commission error, I would say. Now, if I allow 10% of the training data to be omitted, the area predicted present drops to 30%. So I go from 90%, 97% to 34% of the area predicted present. And then the omission rate is actually good. So at 10% training omission, I get 12% testing omission. It's comparable, so it's not outrageous. I will probably use this instead of this. And that is because I downloaded GBIF data. I mean, I took the data off our WordPress site, but it was GBIF data without any cleaning, and I threw it in Maxent, so I know that I have errors. So I know that not all of those presences are good presences. I shouldn't use the minimum training presence. Now, if you have GPS coordinates, if you have high quality data, you should use minimum training presence because you are very, the quality, you are certain that those locations were accurate, so those locations should be predicted present, those training presences should be predicted present by the model. But if you use GBIF data, if you use data that have error, as you assume have errors, I would not make it so restrictive or so inclusive to have all training presences predicted present. Now, this is where you can balance sensitivity and specificity. We talked yesterday about sensitivity, specificity. I tend not to use these, and I explained because I don't have absence data, so I tend not to rely on specificity that much. So that's one important output to take a look at. These are the response curves. So for example, Bio 14 is mostly flat. Bio 8 is definitely flat. So it doesn't matter how, this is a precipitation variable. It doesn't matter how much variation exists in that precipitation variable. The Maxent suitability value doesn't change, so it doesn't inform the model that much. So these response variables, response curves are good to look at. This is keeping all other, so if I look at Bio 1, it's a model that was built with the 19 variables, this real variable, and all other variables kept at their average sample. So it's, these are additional iterations that Maxent runs in the background in order to generate the response curves. So you see one set, and then you see another set, which is created, which is based on simulations or iterations that use just that variable. So in this case, I have a model that was built just using Bio1. Here, I have a model that was built using Bio1 plus all the other variables, but the other variables were kept at their sample average. So differences in between the two. So a model based on one variable, I tend to look at these, not at these. Okay, then the famous variable contribution table. Wow, it's so late. Variable contribution table has most important to least important variables. So this means that Bio 11, Bio 12, Bio 10 are driving, or these first maybe 5, 6 variables are driving the model, because when these variables are included in the model gain of fit or gain of fit of data increases. And it's a cumulative gain. So every time, anyways, it's a cumulative gain, but in general, when we look at this, we want to see a nice spread of values. If we see one variable driving the model, and I've seen that quite a lot of times where one variable contribution is 70% or 80%, something is not right there. Something is fishy, and by that I mean the other variables are maybe not encompassing much variation, or that one variable is correlated with a bunch of these other variables. So I try, if there's one overriding factor or variable, I try to remove that and rerun the models. Now you see a bunch of variables that have very low contribution. So this would be one approach. I run this one model, and then I decide, okay, I'm not going to use variables below, let's say, 5% contribution. So I'm going to run another model that has only those variables, and these variables are removed. There is no rule of thumb in terms of what would be the contribution threshold. Also, I don't think it matters that much when you rerun the models without these variables. Unless these variables sum up to 40% contribution, the model won't change that much. So if these top variables represent 80 or 90% of the contribution to the model, eliminating this 5% cumulative contribution changes the model, but it doesn't change the model that much. Okay, so let's see that model output. So I have an ASCII file. If I remember where I put it. I thought I had it open. Okay, so I have an ASCII file. So basically the potential, the suitability, and it says it doesn't have spatial reference because it doesn't have a projection file. But here is the Maxent suitability map. I'm going to remove this, sorry, that. And it goes from a very low value to a value close to 1, but it will never be a 1. So this is the logistic output. And we can play with the colors. Obviously, we can change that. What is surprising is the number of times we've seen papers that take this little picture here and plaster it on in the paper. And that is so disappointing. Try not to be so I don't know, obnoxious. But yes, this happens so many times. I teach a grad course, like I said. I probably taught it 5 times now. I tell the students, please do not include this in your report. And I have a few. I don't have one. I have a few of, the class is 15, 20 students. I have a few who include this. And I'm thinking, why did we go through all this, learn how to use ArcGIS, look at maps? if you're not looking at the output. The other thing is, this is some sort of scaling of the logistic output. The colors are not very informative, whereas here, I can create my own beans, my own classes, and also what I can do is I can go to the output here, and remember I said if I'm going to use the minimum training presence, I will have 97% of the space of the area predicted present. I'm going to use the 10 percentile, so I'm going to use this value 0.256 to basically create a presence-absence map. So I'm just going to change the symbology here. And it's a large file. It's going to take a bit to create those classes. And I'm just going to do, I think it was 2.56. Oh, sorry. I should do 2 classes first. I think it was 5, 6. Double check. Yes, 5, 6. Okay. Okay, so this looks, at least in my mind, to me, this looks a bit more informative than this. So I tend to look at, or you can create thresholds. For example, I had the continuous output. I could do pixels at which, at the threshold of 10% omission error, 20%, 30%. So I can do that. I can show that if I play with the output in some GIS or in R, but look at the output, not just the picture. And you can also, check the, see, the model wasn't that bad, even with crappy data, but it can get better without those outliers. One other thing I wanted to mention is there are a bunch of other outputs here. So this is the map. This is the HTML, the summary of data, of results and data used. Then we have this, and I think it's okay to just look at it for now like this. So I have the coordinates, longitude, latitude. If it was used test or train, the points, if they were used for training or testing, and then I have the Maxent value associated with each of these points. So that's basically, I should actually just open it in Excel. So remember how I said I should remove remove duplicates. I should do the spatial subsampling outside of Maxent. If I don't do that outside of Maxent and I throw it in Maxent, I get the spatial unique, the number of spatial unique locations that were used in the model to test or train the model. So I have a total of 2,170 presences, and the number of presences. Oh, it actually says here the total number is the sum of training plus testing, which is 2,100. But my original data set had, I think, 4,000 occurrences. So out of I think it was it 4,000? I can't remember. But it was a large data set. So a lot of locations were duplicates, and then I had probably a lot that were not spatially unique at the resolution at which the climate data are, which is 4.5 kilometers. But you can also, I think this is a nice output because you can see which of your locations were used to train or test the model. So you can see the spatial, you can map those points in ArcGIS, or you can display them and plot them in R and see where are the presences that were used for training the model and testing the model. One last thing is, remember I used a threshold in Maxent, so I went to settings, I think it was in advanced, and I said apply threshold rule, and I applied minimum training presence. Now that minimum training present, that threshold generated a second output, a second raster, which is Dasypus, which is the armadillo thresholded. So armadillo thresholded at 0 training omission error looks like this. In a moment. So that's the entire 97% of the space predicted present versus 10% omission error. Just omitting 10% of my data, I get something that I think is, well, it is more meaningful than this. Okay, so the outputs, if you use a threshold, you will have two rasters, the continuous one and the thresholded one. If you do cross-validation, you will have replicates. So you'll have, if you do 10 replicates, you'll have 10 of these 10 Dasypus 10 replicates, 0 to 9. They are numbered. You will also have an average raster, a minimum and maximum value raster, and I think a standard deviation raster. So when you do cross-validation or bootstrap or anything, when you do replicates, you will have multiple outputs because Maxent is generating what you told it to generate with replicates. If you do projections, the name of your projection folder, so let's say I'm training the model in North America and I'm projecting the model to Europe. The name of my projection directory is Europe. When I get the outputs, there will be a Dasypus -Europe or projection Europe, something like that. So make sure you name your projection data set something that makes sense to you, because that will be used to label model outputs. I'll stop now. Yes?
Would you put the map and the occurrence data on top? Yeah, so even though there's no presence in Uruguay, I was really expecting the model to predict Uruguay because the species is really very prevalent and abundant there. So this could be a problem of some pixels having a lot of votes. Because I was disappointed. I was expecting that binary data was gonna predict Uruguay, because it's very similar to the conditions in Argentina there.
I can make it happen.
So please use that data and tell me that Uruguay is gonna be there, because I am concerned.
No, and that's very, very good point. This is a throw data at a model. I didn't clean the data. I didn't make sure that the parameters are optimizing or making the model reasonable. So yeah. So now I'm just playing with this, but yeah. So if we lower the threshold. And this is an example of how you can be misleading with your. So I dropped. Oh, I didn't know you're from Uruguay. Sorry. So this is how to lie with maps. Don't. Unless you have a reason to say, oh, it shouldn't be. So my threshold was based on this, right? So my threshold, this value 2.56 was based on 10 percentile training presence omission error. When I dropped this to 0.1, I increased the omission error, but I don't know by how much. So this is not how you should do it. You should go back. You should actually go to one of the outputs and decide, okay, this is an out. Sorry, I have to open it in Excel. So this is another output. I wanted to give Shao time to talk, so I was gonna stop, but it's a very good point. So I do minimum training presence, and I realize 97% definitely not useful. Then I say 10% omission error, Uruguay is not included. So I need another threshold. So I can go into this Maxent output that shows me at what, it shows me test omission error and training omission error. So I don't want it, let's say I want it to be, instead of 10%, I want it to be 5%. I find the 5% omission error. And so the logistic output is 1 point, I think that's the logistic value. Yes. So at 5%, it should be the logistic value is 169. I used 0.1. So basically, I allowed 1% omission error. Go ahead.
Just to avoid this being a little too much adjusting the map to make sure that Uruguay has armadillos, what you can do more formally is before you start the modeling process, but after you have cleaned up your data a little or a lot, or in this case, not at all.
Not at all, just
Right.
threw everything at the model.
Mona just dumped garbage into Maxent. But what you can do is you can do essentially a priori an assessment of how much error is likely to remain in your location data. If these are all data that the taxonomic specialist checked the IDs and all of the coordinates are with the GPS unit, then you can assume that your error is fairly low. But if this is just dump the garbage out of GBIF into Maxent, then you've got to be thinking of a fairly high level of error, because it could be misidentifications, it could be bad coordinates,
The
it could
centroid
be
of the
all the
country.
stuff that Xiao showed us yesterday with coordinates that look good and really they're crap, right? So what you want to do is you want to do a slightly quantitative but still a bit qualitative evaluation of kind of how clean or dirty are my data. Really, really dirty data, you're probably looking at 5 to 10% error. And really clean data, you're somewhere between 0 and 1%. And you saw that made all the difference in these predictions. And so you would state that assumption about how clean your data are a priori, and you would use that. And in a paper that Mona and I and one other person did in 2008, where we proposed the partial rock approach, we offered a term of capital E for error that is essentially this a priori guess of the proportion of error. And you're going to use that in thresholding here. You're going to use that in the partial rock algorithm for model evaluation. And you can even end up using it in model calibration as well. So, that'll make it a little less, how to lie with maps, although it's quite true,
And it may not be, someone really wanting to lie with maps, but someone saying, well, I know this is, I know the species is present in these 5 states. So something is wrong with my threshold. What the wrong path to take is to find the value that matches what you are looking for, as opposed to decide on the threshold value based on how messy data are. You could if you clean your data and it's perfect, well, as perfect as it can be, and your models are still omitting important big regions, you go to the basic, not advanced, you go to the regularization multiplier and you relax the model fit of data so that more areas are predicted present. So that's
But what you would do here first is
Clean
first
data.
balance the data.
yeah.
I mean, put up the map, Mona, if you would.
Well, I was going to just show you one example here.
Sure, sure.
This is 110 kilometer resolution. It's bogus, and you have 10 kilometer resolution, right? So you have garbage here. You have real garbage.
But probably more.
You have fresh and very stinking garbage.
That's right.
This is fresh garbage. This is stinking garbage. The 100 kilometer resolution. Yes, and then
I'm guessing that the garbage that really, really makes you throw up your lunch is, look at that. You know, probably 90% of the data are in the U.S. and Mexico.
yeah.
And then all of South America, or everything from Colombia south and east, is probably representing 3 or 4% of the data. And so whatever differences there are in climate, those are being left out systematically. And so I'm guessing that if you kind of balanced the northern part of the range down to the densities that you're seeing in the southern part of the range, that probably your model is going to be much more satisfactory, and Uruguay wouldn't have to worry about the extinction of a mammal species. I mean, seriously, that's what's driving this. So don't worry too much about this.
No, the only concern is if the lack of Uruguay being there, it was of the lack of occurrence, or if there's something wrong with the predicting whatever
There's-
you feed into the model.
There's something very wrong.
Between the two.
This is very bad, the clustering of data. Also the fact that I threw very coarse, very large uncertainty into the model. When I show Maxent for the first time in my class, I use a virtual species and it has perfect match. But that's not what we want you to see. What we want you to see is at least my view on teaching you how to use Maxent if you haven't used it yet, is that there are data out there. There's GBIF data. If you misuse it, you get questionable results. So I think we are past even coffee break. I don't even want to check the schedule. It's really bad. Oh yeah, we are 10 minutes past 10 minutes past the break. We didn't do the practice, and I ate 30 minutes of Shao's time. So good luck, Shao, finishing your stuff. But yes, we do want you to at least be aware of many of the pitfalls and limitations, and not make these mistakes when you go home and you start using Maxent.